Okay, welcome to the ninth lecture about cell organelles. Now, this video I'm gonna cover lecture 9, 10 and 11 because all these three lectures are about cell organelles and all of them are given by Professor Peter Nike. The first lecture is really nothing, it's an introduction. Most of it you probably know from before, but the real material is lecture 10 and 11. So let's do this introduction first. As you know, the organelles inside our cells are compartmentalized. It means that we've got organelles, we've got these compartments in our cells, which are, a lot of them are bound by a membrane themselves. So if our cells themselves, they have a cell membrane, why do we need membranes around our organelles? Well, the main reason is that each organelle of the cell is responsible for a specific set of enzymatic reactions or reactions in general. And these reactions, each of them, because they are specific, they need certain conditions. One reaction may need a high pH, another may need a low pH, one may need a high temperature. And if all these reactions were going to happen at once, the cell simply couldn't handle it, because the conditions required for one reaction is the conditions which can inhibit another one. And that is why we've got our organelles compartmentalized. So each reaction will take place in the medium which is perfect for it. And that is the biggest reason that our organelles are bound by membranes, that each of them has a different medium, has a different, provides different situations for reactions to occur. And here is a list of cell organelles. We're gonna talk about almost all of them in the upcoming lectures, but you probably know that some organelles are not membrane bound, like plasma membrane, cytoskeleton, centrosomes. They are not bound by membranes, and by some more strict definitions, only organelles with only membrane-bound organelles are considered to be organelles. But the way we're gonna look at it, or the way the department looks at it, I think, is that we've got membrane-bound organelles and we've got other organelles. So, as we talked about, there is an advantage for these organelles to be membrane-bound, to be compartmentalized. And the best advantage is that providing different situations. But there is also a disadvantage, because in a cell we need transport of material, transport of proteins from one organelle to another. And because these organelles are now bound, are now in different compartments, these transports are usually very complex and they require energy, and that is the disadvantage. Here's also a very good picture which I really recommend just looking at it and kind of memorizing these organelles by their shape. It really helps. It helped me. And the last thing about this introductory lecture is that we divide the cell organelles, or the inside, the cytosol of the cell in general, into different categories based on two points of views, an evolutionary point of view and a transport point of view. From a evolutionary point of view, as we talked about, organelles like mitochondria, they have endosymbiotic origins. So these organelles, like mitochondria, came from prokaryotes. and now here they're presented with red color. And peroxisome is also another organelle which came from a endosymbiotic origin. And between peroxisome and mitochondria, only mitochondria has got DNA for itself. So peroxisomes, they don't have DNAs for themselves. And as you can see here, the space between inner and outer membrane of mitochondria, the intermembrane space of mitochondria, is colored pink, which is the color of extracellular space. And that is because the inner membrane of mitochondria is the membrane which was originally the membrane of the prokaryote, and the outer membrane of mitochondria is the membrane which was originally the cell membrane, the membrane of eukaryotic host cell. So the space which is filled between these membranes is originally was the fluid from outside the cell, was the extracellular space. And now we're talking about the evolutionary point of view. So it's rational that the intermembrane space is pink. And you can see here that the nuclear envelope and also the lumen of ER and the lumen of Golgi complex and the lumen of lysosome are pink in color, which represent the extracellular space. Now these lumens are formed via invaginations of the first eukaryotic cells. So when the cell was invaginating its membrane inside itself to form these structures, to form the nuclear envelope or ER or Golgi, the fluid from extracellular matrix was going through these invaginations and formed these parts. And then we've got the cytosol of the cell, which is a part of the cell which existed even during those eukaryotic stages before engulfment of these prokaryotes and at the very beginning of life. Now, from a transport point of view, we can divide the cytosol of the cell into the endomembrane system transport and cytoplasmic compartment transport. The endomembrane system transport is between ER, Golgi, lysosomes, vesicles like vesicles which are endocytosed to the cell or they're trying to fuse in the cell, and the lumen of ER. And the lumen of nuclear membrane. The transport between these compartments, the transport between endomembrane system, is usually via vesicular transport. So if you imagine this is ER, the transport between ER and Golgi is that a vesicle will form from the membrane of ER, will form like a vesicle like this from the ER, and will fuse to Golgi. And that is how we transport between ER and Golgi. Whereas the transport between cytoplasmic compartments, like the transport in the cytosol and the cytosolic organelles, like nucleus, peroxisome, and mitochondria, is usually via diffusion or directed motion. And that is the second point of view, which is the transport point of view. Now that we know the very basics of cell organelles, let's now talk about each of them in a specific, and let's start with the ER or endoplasmic reticulum. And since, as you probably know, we've got smooth and rough endoplasmic reticulum, we've got smooth here and rough endoplasmic reticulum. We're gonna start with this smooth endoplasmic reticulum. And in case you don't know, rough endoplasmic reticulum is called rough because on the membrane of rough endoplasmic reticulum there are a lot of ribosomes, and these ribosomes under electron microscopy look like the surface of ER is rough, whereas there are no ribosomes on the surface of smooth ER, and that's why it's called a smooth. And we're gonna first talk about the smooth ER. Now, smooth ER has got different functions. One of the important ones is storage and release of calcium, which we talked about in calcium homeostasis lecture. Another one are the biochemical reactions happening in smooth ER. So a general rule of thumb about endoplasmic reticulum in general is that when we're talking about smooth ER and chemical reactions in a smooth ER, we're usually talking about hydrophobic materials. We're usually talking about lipidophilic material or lipids themselves. So we're talking about the reactions of lipids, cholesterols, and steroid hormones. Whereas when we're talking about RER, usually we're talking about proteins. So keep that in mind. It can help you to answer questions without even knowing them in detail. So smooth ER, usually about lipids, cholesterol, steroid hormones, lipid connected things. Whereas rough ER is usually about proteins. But one of the most important things that smooth ER is responsible of is detoxification. Detoxification is a typical reaction, especially for the smooth ER of liver cells. Now, what these smooth ERs of liver cells are doing is that they form water-soluble or hydrophilic material from lipid-soluble or hydrophobic material. And why is that? Because you see, a lot of drugs, a lot of toxic material which we do not need are bound with, for example, serum proteins in blood. The thing you can see here is part of a nephron, is part of kidney. So in our kidneys, basically what happens is that drugs and material which we don't need is going to become part of the urine, so it has to leave the blood circulation. But a lot of these drugs, a lot of these material which we don't need and are toxic are bound within, for example, some serum proteins, and they cannot leave the epithelial cells of the wall of our vessels. So what our body needs to do is to release these hydrophobic toxins from these proteins. And in order to do that, our body needs to transform these hydrophobic toxins into hydrophilic toxins. And that process happens in our smooth ER of liver cells especially. And the enzyme, there are lots of enzymes helping for this reaction. There are actually two phases of this conversion. But one of the most important enzymes, which I think you need to know, is cytochrome P450, which is the most important one. And as I mentioned, there are two phases of reactions. A phase 1, like hydroxylation, and a phase 2 reaction like addition of glucuronic acid. And I do not think you need to know in a specific like what happens in each phase, or even that the phase 1 reactions are monooxidation and the phase 2 reactions are conjugation of hydrophilic groups. But it's good to know these, and it's I think very easy. Now that we're done talking about smooth ER, let's talk about another organelle, lysosomes. Well, lysosomes are also, as you know, membrane-bound organelles, and they're responsible for degradation of material either from intracellular or from extracellular origins. And how do these lysosomes exactly degrade material? Because we have got other organelles responsible for degrading material also. We've got proteasomes, we've got peroxisomes. They are also responsible for degrading material, but the mechanism of that each of these organelles work are different. In case of lysosomes, we have got acid hydrolases inside the lumen of lysosomes, and these acid hydrolases hydrolase material. They hydrolase proteins, nucleic acids, and everything that is mentioned here, which you don't need to know in particular. You don't need to know what the names of each of these acid hydrolases. You just need to know there are different types of acid hydrolases in the lumen of lysosome, and they need an acidic pH in order to be functional. And this acidic pH is provided by a V-type proton ATPase on the membrane of lysosome, which transports protons or hydrogen ions inside the lumen of lysosome and makes the lumen of lysosome acidic. And if you remember, we talked about these V-type ATPases and other type of ATPases in previous lectures. So now that you know how the lysosomes work, you probably should guess what happens if these lysosomes do not function. Well, storage diseases will happen. Storage diseases are due to inherited lack of certain types of lysosomal hydrolases. So it's inherited from one generation to another. It's congenital. And what basically happens in case of storage diseases is that some hydrolases, as I said, are deactivated. So the material, for example, let's say proteases are deactivated. So proteins in the lumen will accumulate because they cannot be hydrolyzed, and that will cause the swelling of lysosomes, and that swelling will eventually cause the whole cell function to be disrupted. An example of storage disease, which is good to know, is Tay-Sachs, and I think you need to know at least two examples. You can memorize Tay-Sachs, and you can also memorize Fabry disease, for example. The examples are in your minimals. You can look at them later. So, before I talk about autophagosomes, which are related to the lysosomes and what happens, first you need to know something else, is that we've got mainly two types of lysosomes in our cells: primary lysosomes and secondary lysosomes. Primary lysosomes are just lysosomes with all the acid hydrolases inside them, but without the material to be digested. So the only difference between primary and secondary lysosome is that secondary lysosomes are lysosomes with the material to be digested inside them. And that is primary and secondary lysosomes. Now let's talk about autophagosomes and autophagy in general. So what is autophagy in general? It is when a cell degrades all its components in its lysosome, and that is important. For example, when a cell is very old, that has lost most of its function, so body needs to get rid of this cell. The process is called senescence. So, for example, in senescence, we've got a membrane-bound organelle called autophagosome, which, when this process is initiated, will fuse to lysosome. So autophagosome will fuse to lysosome and will form autolysosome. And now these autolysosomes are responsible for degrading cell organelles. So now that we talk about lysosomes, let's now talk about peroxisomes. As I mentioned, peroxisomes are also responsible for decomposition of material, and they are found in great number in liver and kidney cells. But the way peroxisomes work is that they have some enzymes like peroxidases. So peroxidases are the most important enzymes of these peroxisomes, and these enzymes use an oxidative decomposition of material inside these peroxisomes. And when we say oxidative decomposition, the consequences of these decompositions is that we will have some new species of molecules that are toxic, that we don't need them as side products of these decompositions, like hydrogen peroxide. So as a consequence of action of peroxidase, which is an important enzyme of peroxisome, we will have hydrogen peroxide as a side product. And well, this hydrogen peroxide is very toxic, and the cell needs to get rid of it as fast as possible. So peroxisomes will create another type of enzymes called catalases. And catalases are responsible for neutralizing these hydrogen peroxides and to neutralizing them into water. Now that you know about peroxisomes, here there is a nice chart comparing mitochondria, lysosomes, and peroxisomes. Well, first of all, let's compare lysosomes and peroxisomes. And since we said lysosomes, they do hydrolytic degradation with these acid hydrolases that they have, whereas peroxisomes undergo oxidative degradation. So this is the main difference. And the similarity is they both degrade material. But the difference between mitochondria and peroxisome is that, if you remember, mitochondria also has got some oxidative degradation reactions. If you remember, oxidation phosphorylation occurs in mitochondria, which is as the result of TCA, the NADH molecules will be the electron carriers, and the electron will then be transported in the electron transport chain, and the last electron acceptor in the electron transport chain in mitochondria is oxygen. But the difference between mitochondria and peroxisome, the comparison here is very good. Mitochondria is like the nuclear power plant of our cells. So the energy of this electron can be stored as a form of ATP. And at the very end, this oxygen, which is the last electron acceptor, will become water directly. Whereas in peroxisome, it's like a nuclear bomb. There will be no energy stored. It will be used in order to degrade, to destroy materials inside the peroxisomes, and that the oxygen, which is now accepted, that electron, will be converted to hydrogen peroxide, and we will need catalase to neutralize this hydrogen peroxide to water molecule. So now let's talk about rough endoplasmic reticulum, which, as we said, is mainly responsible for modifying proteins. But first of all, we should know that how these proteins end up in the lumen of RER. Can any protein just be transported into the lumen of RER, or is there a specific transport? Well, there is a specific transport, and not any protein can be transported to the lumen of RER. Proteins which are destined to end up in the lumen of RER, they need to have a special amino acid sequence on their N terminal, a signal ER signal sequence, which is the sequence of 15 to 30 amino acids and is usually hydrophobic. And as you can see here, this gray structure here is a ribosome. And as ribosome is reading the mRNA, it's producing the polypeptide chain, the protein here. And at the very end terminal of this protein, which is getting translated, which is just getting generated, at its end terminal, there is a signal sequence. There is an ER signal sequence. And there are particles in the cytoplasm, signal recognition particles, these green structures. These particles will recognize the ER signal sequences and will bind to them and will transport the whole protein with its ribosome at the same time that the protein is getting translated to the docking site, to the site where this complex will dock with the membrane, to the membrane of RER. And I do not think you need to know the translocon or these different subunits. This whole part is the docking site for signal recognition particle and the peptide itself. And when that happens, the ribosome will continue translating the protein to the lumen of ER via these translocons. As I said, I don't think you need to know the name of translocons. So via specific type of openings, these ribosomes will continue translating or producing their proteins into the lumen of RER. And when when the protein is moving into the lumen of RER, when it enters to extent that its whole entrance is ensured, a special type of enzyme, a signal peptidase, will cut off the signal sequence, will cut off the ER signal sequence, because we don't need this signal sequence anymore, because the polypeptide is now ensured to be in the lumen of ER. And as you can see here, and as you can obviously conclude, these proteins are transported to the lumen of ER in an unfolded form. And it is after their transport that they will go back to the folded functional protein. What happens now is that, as we said, these post-translational modifications for the protein in the RER will occur. One thing before I start talking about what happens to now this fully modified protein is that, if you remember, as we said for mitochondria, because in case of mitochondria, proteins are also transported in unfolded form. When these proteins are transported, before they go back to their folded three-dimensional form, there are a special type of proteins, heat shock proteins or chaperonins, which prevent the incorrect folding of these proteins. So these proteins will go back to their fully folded form with help of chaperonins or heat shock or HSP proteins. That is good to know. And when and only when these proteins are fully modified and completely ready will they leave the ER to their next destination, which is Golgi complex. But as we said, only when they're ready they can leave ER. And in order for ER to ensure the quality of this protein, there is a quality control in ER. So what makes the proteins, those proteins which are not ready not to leave ER? The quality control. And the quality control will give the ER more time to correctly modify the protein. And if these proteins cannot be modified, if the mistake was so big that they cannot be fixed, they will be degraded. Instead, these proteins, instead of getting transported to Golgi, they will be transported to proteasome and will degrade it there. And with this quality control, cell will make sure that wrongly folded, misfolded proteins will never leave for Golgi complex. And when they go to Golgi, when proteins are transported from the ER to Golgi, their journey is just begun, because there will be further post-translational modifications, and at the end there will be sorting of these proteins. And even when proteins which are not meant to be transported to the Golgi somehow are transported there, proteins which are meant to stay in the ER, they will be sent back to ER. And how is that possible? That is because these proteins which were not meant to leave ER, they had some specific signal sequences on them, K-del sequences, which these sequences are actually like a label which says these proteins belong to ER no matter where they are. So even if they somehow manage escaping ER and going to the Golgi, Golgi will find them because of this label, because of K-del, K-D-E-L, and Golgi will send these proteins back to ER. In that form will make sure that nothing which is not meant to be in ER is there. And now let's talk a bit about Golgi apparatus itself. It is located usually near the nucleus, and it's composed of flattened membrane discs, as you can see here, which are called cisternae. So these flattened membrane discs are called cisternae, and as you can see, they're kind of like stacked upon each other. What makes the Golgi apparatus unique, what is its special character, is that it has got a compartmental organization. So there are different compartments within this organelle. These compartments are called the cis Golgi network, trans Golgi network at the very ends of the Golgi complex, and cis, medial, and trans cisternae. Each of these compartments, they have specific enzymatic reactions happening in them. So it's not like this whole structure, no matter where the protein goes, just the same thing happens to it. In different compartments of this Golgi, different things are happening due to different enzymatic activities. What things exactly are happening, like removal of monos or addition of glucuronic acid, I do not think you need to know them. I've never seen them asking in a specific what happens, for example, in medial cisternae, but they like to ask about what happens in trans Golgi network. The trans Golgi network is the only important part of Golgi that I've seen they asked in detail, especially they asked about the sorting of proteins in trans-Golgi network. But one of the things that these modifications of proteins, which is very important, that happens both in RER and in Golgi, is glycosylation, and that is a type of modification that they somehow like asking about. Now, glycosylation basically means addition of oligosaccharides or monosaccharides to proteins. And we have two main types of glycosylations. We've got N-linked glycosylation or O-linked glycosylation. And you need to know the difference between N-linked and O-linked glycosylation. Now, the N-linked glycosylation is addition of oligosaccharides to the asparagine amino acid residue in a protein, whereas O-glycosylation is the addition of hydroxyl bearing amino acids like serine, like threonine, like hydroxylysine. That is, I think, why the O stands for OH, the hydroxyl group bearing amino acids, to the addition, O-glycosylation is the addition of monosaccharides to those amino acids which I mentioned, serine, threonine, and hydroxylysine, because they have a hydroxyl group. So now let's talk about the trans-Golgi network that we mentioned. What happens in trans-Golgi network is classification of proteins that reach there. That what protein is needed to be secreted right away, what protein needs to stand in the Golgi for a while and then secreted when it's needed, what proteins are needed to be always in the Golgi because they are the innate proteins of Golgi itself. These classifications happen in the trans-Golgi network, and it's called protein sorting. So protein sorting happens in trans-Golgi network, and some proteins, they bear a specific type of signal, which is Golgi retention signal. And when proteins have Golgi retention signal, they can never leave Golgi because they are Golgi resident proteins. They are needed for the Golgi itself. Golgi does not secrete them. But in the other hand, there are proteins which Golgi needs to secrete. And that is basically what Golgi does, right? Because we've got proteins getting their main modifications in RER, then they're transported to Golgi. So Golgi kind of packed them and labeled them to where and when these proteins need to be secreted. And out of these proteins which are secreted, because as we said, there are some proteins which will remain in Golgi. So first of all, there are two main types: proteins which will remain in Golgi, proteins which will leave Golgi. Out of the proteins which will leave Golgi, we have two types of secretion. We've got constitutive and regulated secretion. Well, constitutive secretion is the exocytosis of without any delay, which is happening always, which is always happening, like the extracellular matrix protein, which is a good example. Cells need to always secrete the extracellular matrix protein without that much of a regulation, and that is happening in the trans-Golgi network via the constitutive secretion. Whereas some other material, their secretion is needed to be regulated. Like, for example, hormones. Hormones are a good example of a material which are needed, which their secretion is needed to be regulated. And these are two main types of protein secretion from Golgi. So when we're talking about all these transports that's happening in the cytoplasm of the cell, we said that these vesicles will fuse with these ones, and these vesicles will fuse with the membrane, with the cell membrane, in order for exocytosis, for example. Now, how do these vesicles recognize each other? Like, how does a vesicle know exactly which one of all these different types of vesicle in the cell to fuse with? That is not random, of course. There are special proteins, snare proteins, SNARE proteins, which are present on the membrane of vesicles, and they're responsible for the specificity of the membrane fusion. And since we've got a vesicle and a target membrane from another vesicle, or the lumen of, for example, Golgi or somewhere else, we've got two types of snares: a V snare, which is on the donor vesicle, and a T snare, which is on the target, on the membrane of target vesicle. This picture here is better. So we've got V snares on the membrane of donor vesicle and T snare on the membrane of the target vesicle. And these proteins, in order to bind, they need some adapter proteins to facilitate their binding, which these proteins, which I'm talking about, are SNAP proteins. You don't need to know like different types of SNAPs, like alpha SNAP, NSF, just know SNAP. So T snare will bind with V snare, which is responsible for the specificity of vesicle fusion, and SNAPs help this whole process. Now that we're done with those organelles, which are important for you to know, we can now talk about how material gets into the cell. We can talk about endocytosis. Now, endocytosis, the definition, is uptake of material from the extracellular space by enclosing it into a membrane vesicle. So this vesicle is part of the cell membrane, which will become separate from the cell membrane to form the vesicle. And we've got two main types of endocytosis: pinocytosis and phagocytosis. Pinocytosis is also referred as cellular drinking because it's happening because pinocytosis is happening constitutively. It's happening without that much of an effort for the cell. And pinocytosis is the uptake of dissolved material. So it's the uptake of usually hydrophilic material, whereas phagocytosis is the uptake of like these big particles, and that is like one of the differences. You can see pinocytosis, the uptake of very small particles, like when you need to drink, and phagocytosis is the uptake of big particles, like when you need to eat. And then pinocytosis itself will divide into different types of pinocytosis. It will have subcategories. But one thing to mention is that the subcategories that the department gives is not always the categories which are in the book or you can find in other places. So it's always better to, if you're looking for a category, like what are the different categories, it's always better to look in the lectures for cell biology rather than looking in the book. Now, a specific type of endocytosis which in the lecture slides is part of the pinocytosis, is called receptor mediated endocytosis. So what receptor mediated endocytosis is that only the material specifically bound by cell surface receptors is taken up. So the thing for receptor mediated endocytosis is that we've got receptors. Whereas, for example, when we're talking about pinocytosis or phagocytosis, the material comes, will be in the vicinity of the cell membrane. Cell membrane will invaginate, will form a vesicle, and the material will be in the vesicle. But there are no receptors in case of the normal endocytosis, the normal pinocytosis. Whereas there are receptors on the surface, on the cell membrane for receptor-mediated endocytosis. And there are different types of receptor-mediated endocytosis. Clathrin-dependent, caveolin-dependent, clathrin and caveolin-dependent. You don't need, as far as I know, you don't need to know anything about caveolin-dependent endocytosis, but you need to know about clathrin-dependent endocytosis. Now, what is clathrin itself? Clathrin itself is a structure with 3 heavy chains and 3 light chains, which these each of these monomers, which each monomer has got 3 light chain and 3 heavy chain, will form pentagons and hexagons. And this is also written in your minimals, but it's really hard to understand what we're talking about. So I found a very good video on YouTube. Here, this one.
The functional unit of clathrin is known as a triskelion, a 3-legged structure made up of 3 heavy chains and 3 light chains. Extended interleg contacts between different triskelions allow clathrin to self-assemble.
So, as you can see, each of these ones you saw at the beginning are one triskelion clathrin with 3 heavy chains and 3 light chains. And when they form a kind of a lattice like this around each other, they form the hexagons and pentagons which we talk about. And in this lattice, within this structure, there is our vesicle. So what happens exactly for the formation of this clathrin coat is at first you will have the binding of the cargo or the ligand with the special receptor, with the specific receptor on the cell membrane, because we're talking about receptor mediated endocytosis. Because only in case of receptor mediated endocytosis we've got these clathrin, the formation of these clathrin coats. We don't have the formation of clathrin coats in case of, for example, phagocytosis. But in receptor mediated endocytosis, when the ligand or cargo binds to the receptor, the formation of clathrin coat will be initiated, and clathrin coat will be formed, as you can see in here, will be formed in a way to form a vesicle from the cell membrane. And clathrin will not directly bind to the receptors. Clathrin will bind to receptors via special adapting molecules. You can see, for example, here it's AP2. You don't need it's called AP2. What you need to know is that these are adapting molecules, adapting proteins. Sorry, that clathrin coats are formed with help of these adapting proteins. And when the clathrin coat forms most of this vesicle, when it wants to cut the membrane and form the vesicle, as you can see here, when the clathrin, these dark green structures are formed all around this part of the cell membrane, and now we need to cut the cell membrane. We have dynamin proteins helping the neck of this vesicle to be cut and the vesicle to be formed. So dynamin and adaptins, these lightish green structures, are important for the exams. They usually ask about dynamins. And when everything is done, when the vesicle is formed, these clathrins will leave the vesicle, will detach from the receptors in order for the vesicle, for the snare proteins of the vesicle to be exposed. And why the vesicles need these snare proteins to be exposed? In order that so they can fuse with the vesicles or the organelles that they are meant to fuse with, because we entered these ligands into the cell for a reason, right? We want to either degrade them or use them in different manners. Now, there are I think three types. There are three examples of endocytosis which are important and you need to know about. And what you need to know about these examples is only that what happens to the ligand and to the receptor at the very end of receptor-mediated endocytosis. And the first one is the endocytosis of LDL particles. So this endocytosis is a receptor-mediated endocytosis because we've got receptors. Now these yellowish circles are LDL particles. They will bind to their LDL receptors and the receptor-mediated endocytosis will be initiated. The formation of vesicles will happen, and in case of receptor-mediated endocytosis of LDL, we will have degradation of LDL of ligand itself, but receptor will not be degraded. The LDL receptor will goes back to the cell membrane. So we will use a receptor several times, and that is for the LDL. Whereas, for example, in case of transferrin, there is a different story. When transferrin, which is the transferrins, the molecules are bound, the proteins are bound to their receptors, and the receptor-mediated endocytosis occurs, not only the receptor will not be degraded, even the ligand will not be degraded. So everything will just goes back. What happens is that transferrins are for the transfer of are meant for the transfer of iron. So they will lose these irons inside them when they are in the vesicle. But transferrin itself and its receptor, nothing happens to them. So in case of receptor-mediated endocytosis, the ligand and the receptor, none of them will get degraded. They will both get back to the cell membrane. And in our last example, which is receptor-mediated endocytosis of EGF, both the ligand, which is EGF, and the receptor, which is EGF receptor, both of them will get degraded. So these three examples, what you need to know is that there are three, all of them are receptor-mediated endocytosis. In case of LDL, ligand gets degraded, receptor doesn't get degraded. In case of transferrin, ligand and receptor, none of them gets degraded. And in case of EGF, both of them get degraded. And you almost know most of the things that you need to know about this lecture, except transcytosis. Well, transcytosis is when endocytosis happens. It is when a vesicle is formed when the endocytosis happens, but nothing will happen to that vesicle. So that vesicle will exocytose from the other side of the cell. As an example, the vesicle will enter the cell from the apical domain and will leave the cell from the basolateral domain of the cell. So some proteins will come in some vesicles and they will leave from the other side of the cell. Nothing will happen. That is transcytosis. Endocytosis we talked about, and exocytosis we also talked about in other lectures. But again, exocytosis is very easy. It is when the material in the vesicles are released to the extracellular matrix with the fusion of the vesicle to the cell membrane. So the exact opposite of endocytosis. And that was all about this lecture. See you in another one.